ആ അങ്ങുണ്ട്. ഞാൻ ചെന്നോ പറ്റിയിട്ടുണ്ട്. Hello, good evening all of you. Welcome to our session. Last time we were discussing the classical blood group or ABO blood group discovered by Landstein, Levin and von Castello. Von Castello is the man who discovered the AB group. Actually, A, B and O were first discovered. AB was discovered later. That is why this group is called ABO group, classical blood group. Okay, do you have any doubt on that? Anybody? Anybody having a doubt on that? I hope all of you are having this chart. So, in that connection, I told you O group is called universal donor because he is having no antigen. That is the reason. And AB is called universal recipient because he is having no antibody. So, in blood transfusion, though reaction occur between antigen of the donor blood with the antibody of the recipient blood. That is more important. Reaction also occur between the antigen of the recipient blood with the antibody of the donor blood. But that is not clinically important because we are getting only small, little antibody containing plasma through the donor blood. So that reaction is not important. What is more important is antigen of the donor blood reacting with the antibody of the recipient blood. Okay. Now, almost 40 years after that, 40 years after that, two scientists, they were studying the histocompatibility, that is blood tests between different animals to show whether they are related or not. In one of the studies, Levin and Fisher, they are the two scientists who studied these things in detail. In 1940s, they took the blood from a monkey, rhesus monkey, and gave it to an animal like rabbit. So what happens? Normally, rhesus monkey blood given to rabbit. They are not at all related to each other. And so, the rabbit will be developing antibodies. It will produce antibodies against the antigen of the rhesus. Antibody against rhesus antigen. And to this rabbit blood, which is containing antibody against Rh antigen, they mixed blood of so many animals. And in one of their experiment, they added in this blood, antibody containing blood of rabbit, they added human blood also. And very often the human blood clotted. Clotted. What does it shows? This antibody against Rh antigen is present in the blood. That is reacting with the human blood means human blood will be having the same antigen as is present in this rhesus. That's why reaction is occurring, because the antibody is against the Rh antigen, and here human blood is clotted because human blood is containing the same antigen that is present in rhesus monkeys. And thus they understood that most majority of the human population, more than 80%, actually it is even more than 90% of the human population are having the same antigen as is present in rhesus monkey. And the antigen present in rhesus is called Rh antigen. And if the same antigen is present in human beings, that means they are having Rh antigen. Those having Rh antigen are called Rh positive. So more than 80% of the human population are Rh positive. And very rarely, some persons, less than 20% or actually less than 10% are having no such antigen. So it is called to be Rh negative. This Rh antigen is also present on the surface of RBC and is an entirely different antigen, different from ABO antigen. What is the unique feature, the most important feature of this Rh blood group? So people can be classified into two. Those which are having Rh antigen, they are called Rh positive. Those which are having no Rh antigen, they are called Rh negative. Then, most important point is that a man who is having no Rh antigen at all, that is Rh negative, will be having no no antibody also against the RH antigen. The RH antigen against it, no naturally antibodies are present. By nature, such persons are having no RH antibody. But you can induce RH antibodies. You can make them produce RH antibody when the RH positive blood is introduced into their body. Suppose RH positive blood is introduced into an RH negative. Then this is a new antigen. It is a new antigen to their body, and their body will respond, their body will react, produce anti-RH antibodies. So they will be producing antibodies, RH antibody, which is called anti-D. It is called anti-D. Naturally, there will be no RH antibody, but RH antibody will be produced in a negative man, RH negative man, when he is exposed to RH antigens. That is the main difference. Okay. In AB blood group, what we have studied? A man having no RH, no antigen at all, that is O group, will be having both antibodies. But here, he will be having no antibody against the RH antigen. But antibody against RH antigen, that is called anti-D, can be produced in his body. Okay. When he is exposed to RH antigens accidentally. And this is very, very important in blood group transformations or blood transfusions. When we are taking blood from another man, we have to check whether it is AB or O, then positive or negative. A positive man can receive both positive blood and negative blood. Okay, as per the norms. But a negative man can never receive, say for example, he is A negative, he can receive only A and AB. He can receive only A as per our order we are having. See, A group, he can receive A and O, which are all negative. He cannot receive A positive or O positive blood. What happens when he is receiving a positive blood? Since positive blood is having RH antigen which is new to his body, he will start producing slowly RH antibodies. And when RH antibodies are produced, the reaction between the antibody, RH antibody and RH antigen in his body cells will be, blood cells will be occurring. That is resulting in clumping. That is very, very fatal. So, you have to check whether the blood groups are matching or not. Only matching, that is, positive man can receive both positive and negative, because in negative there is no antigen, so there is no such problem. But a negative man can receive only negative. So this is also to be looked when you are receiving blood or donating blood to another person. So in blood transfusions, both these two blood groups are important. Now, second one, This blood group is also important, and this is the explanation why in some cases some babies are having jaundice-like symptoms when they are all born, or sometimes they are all aborted, and the child will be having problems in liver and his blood will be crumbled or will be agglutinated. Such a symptom is called erythroblastosis fetalis, where the fetus will be having more erythrocytes, erythroblast than erythrocytes. Erythroblasts are immature RBCs, and mature RBCs are all completely broken and their number is very, very less than normal. So, abnormal rise in erythroblast, that is called erythroblastosis fetalis of fetus. And this disease is nowadays called hemolytic disease of newborn or hemolytic disease of the fetus. If the baby is born live with jaundice-like symptoms, it is called hemolytic disease of the newborn, HDN. And if the baby is aborted and is coming out as dead baby with problems in liver, problems in blood, etc., then it is called hemolytic disease of the fetus. What is the reason? We are having an explanation. This occurs only when the husband and wife are belonging to two different blood groups regarding RH. The mother is RH negative and father is RH positive. This symptom or this problem occur only in this condition, so that they are having their first child. First child will be RH positive. When the husband is positive or mother is positive, the baby will be naturally having more chance of becoming positive. Okay, it is genetically controlled. So, first baby will be RH positive. And there will be no problem for the development of the first baby. But at the time of delivery, you know that we are having a type of placenta where bleeding will be definitely occurring at the time of delivery. When placenta is separated, baby comes out, then placenta comes out. When placenta is separated from the uterus of the mother, there will be definitely a chance of fetal blood coming out of the placenta, and that will be absorbed at least to a smaller extent into the mother's circulation. Through the uterine wall, this blood, fetal blood that is coming out because of the breakage in the placenta, that will be entering into the mother's blood. So, since the baby is RH positive, the antigen, RH antigen, enter into the blood of mother. Mother is RH negative. So, some of the cells which are from the fetus entering into the mother will activate her immune system, and now she starts producing antibodies. She is producing anti-D. Slowly, it will take some months, she is producing antibodies. So, by the time the first baby is out, and there is no problem for the first baby. So, erythroblastosis fetalis occur, or HDN or HDF occur only in this condition. Mother is RH negative, father is RH positive, and the first child is RH positive. See, first child should be also RH positive. Suppose the first child is RH negative, there is no problem for the second child. The subsequent deliveries after an Rh positive child. Subsequent deliveries after the first Rh positive child. Suppose the second child is Rh positive, then for the third delivery, if there is a third delivery, if the first child is positive, for the second delivery. You got that point? So, the next delivery after a Rh positive child. So, if the first child itself is Rh positive, the mother is getting Rh antigen through the placenta, through the uterus, when the placenta of the first baby is separated, and that causes anti-D formation, and that anti-D is now present in her blood. Now, suppose she is again becoming pregnant for a second time, and this time also the baby is Rh positive. Now, the anti-D present in mother can cross placenta. It can cross placenta. It can come through the placenta to the baby circulation as the baby is developing. Okay, so anti-D will be reacting with the antigen, with the RBCs, and will cause clumping or coagulation. So clumping occur or coagulation occur for the blood. That will be resulting in the breaking down of mature RBCs, and erythroblasts will be remaining, because erythroblasts are not mature cells, they are not having Rh antigens. Rh antigen will be present only in mature RBCs. So only when they become mature, only in mature RBCs there will be a problem, and all mature RBCs may become clumped, and immature erythroblasts will be available, and their number will be more than what is normally present. That is why it is called erythroblastosis fetalis. So this is the situation. Okay. So what is to be done? When the mother is found to be Rh negative, the hospital authorities, health authorities will give an injection immediately after the first delivery. After verifying that the first baby is Rh positive, if the first baby is Rh positive, immediately the mother will be given counter molecules that will suppress the formation of anti-D. The mother is given a Rh antibody preventing anti-Rh that will be preventing the formation of the anti-D. It is against formation of anti-D. It is called Rogam. That injection is given immediately so that it will be destroying all the Rh positive cells that have entered into the mother's blood, so that mother's immunological system is not sensitized, it is not activated, and so no antibody will be produced. So mother, even if she is getting some Rh antigen through the RBCs of the first boy or first child, they will be all destroyed by this one anti-Rh antibody. It can be also described like that. It is anti-Rh. Anti-Rh is the anti-D. So it is anti or against the anti-D. So it is anti-Rh or anti-D or against the antibody formation of anti-D. anti-D is the antibody of RH. That will be prevented. Formation of such anti-D will be prevented if you are given such an injection. Is that point clear? So, this incompatibility, mismatching occur only in this condition. What is the condition? Mother is negative, father is positive. Suppose mother is positive and the father is negative, again no problem, because even if the baby is positive, see, any one of the parent is positive means the most likely that their children are also positive. So here father is positive, that is why the baby became positive. If the mother is positive, the baby will be again positive. And father is negative, the problem is because this RH positive, mother is also RH positive, no problem. If both of them are RH negative, then the baby will be likely to be RH negative, and again there is no problem. If both of them are RH positive, father and mother positive, no problem. So problem might occur only when the mother is RH negative and husband, male partner is RH positive and the first baby is RH positive. Problem arises for subsequent babies, subsequent deliveries, subsequent conceives or contraceptions, for the subsequent deliveries after the first RH positive baby. After the first RH positive baby. So this is erythroblastosis fetalis, or which is also called okay. So the mother starts preparing antibodies against RH antigen in her blood. In case of her subsequent pregnancies, RH antibodies from the mother can leak into the blood of the fetus. How? Through the placenta, it will be coming. Leaking means crossing through the placenta and destroy the fetal RBCs. This could be fatal to the fetus or could cause severe anemia and jaundice to the baby. This condition is called erythroblastosis fetalis and can be avoided by administering anti-RH antibodies. Okay. So, it is against anti-RH antibodies. Okay. Anti-RH. Okay. To the mother immediately after the delivery of the first child. That is called rogam. Okay. Now, we are going to another important point. Any doubt up to the blood groups? Anybody? Okay. We are going to coagulation of blood. What is coagulation? Coagulation means when blood is flowing through the blood vessels, no problem occur. But when blood comes out through small wounds in the blood vessel, the blood oozes for some time, then slowly it comes to a stop. We can see that there is a scum, a cushion-like structure or a sticky structure developing at the site of the wound that is sealing that wound and is responsible for stopping of that bleeding. This scum formation, or this healing of the wound immediately is because of a long chemical reaction occurring, which is a classical example for what is called a cascading reaction. What's a cascading reaction? A reaction in which there are so many steps, and in each step the strength of the reaction, that is number of molecules that are taking part in the reaction, is increasing manifold. Normally, say for example, there are 10 reactions in the first step, it becomes 100 in the second step, 1000 in third step, like that. Number of molecules, the strength of reaction goes on increasing in every step. That is what is called a cascading reaction. So blood clotting is a long metabolic process because of so many chemical reactions, which are all classical example for a cascading reaction. And we have studied that what is the blood cell that is responsible for this blood clotting? Platelets, thrombocytes. Okay, now let us see what this coagulation is. Coagulation is a mechanism of our body to prevent the excessive loss of blood from our body. Suppose a wound is occurred and blood is coming out without any stop, then your body is losing a lot of blood, which is very, very dangerous for our survival. So it should be prevented, that bleeding should be prevented. Bleeding is called hemorrhage. That hemorrhage, bleeding should be prevented quickly. That is a defense mechanism, a protective mechanism of our body to prevent the loss of blood from our body. It is a preventive mechanism, defense mechanism, involving large number of chemicals, and is a classical example for cascading reaction. I will explain the reaction in detail, so please take note of it. Blood clotting occur as a result of a wound. When a wound is created, the tissue of that region, the tissue in the blood vessels, etc., are damaged. Okay. When a wound is created, a wound is formed, the wound will be causing highly serrated margin, uneven margin on the blood vessels, and the blood is coming out. Normally, the inside of the blood vessels are very, very smooth. That is why blood cells are not breaking. But when it comes out through the wound is having serrated margins. It is having very uneven margins, broken margins. So when the blood is coming out through that, the platelets become broken. Okay, and it releases certain enzymes. What are the enzymes? It is called thromboplastinogenase. Similarly, the damaged tissue as well as platelets are producing another inactive material. We have studied that all the elements needed for blood clotting are present in the blood itself in an inactive form. I have asked you to underline that sentence. In an inactive form. So, they are all producing another substance called thromboplastinogen. It is called thromboplastinogen. It is a gen. Gen means inactive. This is an inactive substance produced by the tissues, and this is the enzyme that act on this thromboplasmogen, so that thromboplasmogen is converted into an active form called thromboplastin. See, this is a gen, plastinogen. Gen is an inactive. Okay, this inactive substance is converted into an active compound called thromboplastin with the help of thromboplasmogenase enzyme, which is also produced by the platelets. Is that point clear? Thromboplastin is also called thrombokinase. It is also called thrombokinase, which is an active material. This active material now will be acting on a protein produced by liver. So liver is producing, in presence of vitamin K, a protein which is called prothrombin. Listen, prothrombin, it is a pro means inactive. It is also inactive. It is produced by the liver and is produced under the influence of vitamin K. So vitamin K is highly essential for the formation of prothrombin. It is an inactive material. Prothrombin is now converted into active material called thrombin. And this conversion is occurring under the action of the enzyme thromboplastin, which is also called thrombokinase. For this reaction, calcium ions are very important. Calcium ions are very important for the conversion of prothrombin. So without calcium, clotting will not be occurring properly. So prothrombin is now converted into thrombin by thromboplastin. Now, thrombin is an active compound that will be acting on another inactive material present in the blood, namely fibrinogen. Fibrinogen, it is a gen. Gen means inactive. Remember, all of them are in the inactive form in blood. It is activated by thrombin to active fibrin. Inactive fibrinogen. Remember, we are having three main proteins in plasma: albumin, globulin, and fibrinogen. This fibrinogen is converted into active fibrin. This active fibrin is now polymerizing. Fibrin is a small protein. Once it becomes polymerized, it becomes long chains. So polymerized to threads. It forms fibrin threads. Fibrin is then polymerized to form long threads. Where are the long threads produced? These are all reactions that are occurring in the wound. So you are having fibrin threads. As more and more fibrin threads are produced, they will be all forming a meshwork or a network-like structure. See, these threads are now present in the form of a net, a fine net. As more and more threads are formed, the size of the mesh becomes smaller and smaller. So they will be forming a meshwork whose size, the net size, will be becoming smaller and smaller. So blood has to come out through the gap in between this net. ആ വലക്കണ്ണിയുടെ ഇടയിലുള്ള ഗ്യാപ്പിൽ കൂടെയാണ് ഇനി ബ്ലഡ് വരേണ്ടത്. Blood has to come through the small gap between the meshwork of fibrin threads, which are very smaller. So only liquid part will come. The blood cells, corpuscles are not capable of coming because corpuscles are remaining entangled. Corpuscles become caught up in this meshwork. They become trapped in that meshwork. Can you get it? The blood cells are trapped in, they are all caught up in the meshwork. So, in this meshwork, you will be having blood cells trapped, and the trapped blood cells plus meshwork together will constitute clot. Thus, the clot will be completely sealing the wound, completely covering the wound, and thus it seals the wound and prevents clotting. Only small amount of liquid, a colorless liquid or straw colored liquid will be coming out. What is that? It is plasma. Blood cells are not coming out because blood cells are large in size. They will be trapped in the meshwork, and it is that meshwork plus blood cells, entangled blood cells together, that is constituting the clot. And through the clot, initially there may be small amount of that plasma. So all the proteins are all now used up. Mainly the fibrinogen are used up, and other proteins in the blood are used up for clot formation. So plasma minus clotting factors. Plasma minus clotting factors are coming out. So that is called serum. So in that book earlier, I have told you that there is a sentence. Plasma, so please write it. Plasma without clotting factors. Clotting factors are all used for clotting. What is remaining will be coming out through the wound for some time. That clear or straw colored fluid is called serum. So this is a long metabolic process involving about 13 important factors. I will tell you the first 4 important factors. The number 1 important factor is the fibrinogen. Number 2, prothrombin. 3 is thromboplastin, and 4 is calcium. So these are all essential. A total of about 13 elements are involved in this reaction. Okay. 13 factors in the blood are involved in this reaction, of which the first 4 alone you have to study. Any doubt up to this level? Ananya, Swati Krishna, anybody? Abhinav? So please take down these points. Okay. No such details are given in your NCERT. Clotting is a mechanism to prevent the excessive loss of blood from body. So that is why it is called as a defense mechanism for protecting your body from loss of blood. A dark reddish brown scum, a thick cushion-like structure formed at the site of a cut or a wound injury over a period of time, that is called a clot. It's a clot or coagulum formed mainly of a network of threads called fibrin threads, in which dead and damaged formed elements of blood are trapped. So how the fibrins are formed? Fibrins are formed with conversion of inactive fibrinogen by enzyme thrombin. Thrombin in turn are formed by another inactive substance present in plasma called prothrombin. Okay, so this is what is described from the beginning in your book. It is given in the reverse direction, but I have given it in the forward direction. Okay, there is a enzyme complex called thrombokinase which is required for the above reaction. This complex is formed by the series of linked enzymatic reactions cascading process involving a number of factors present in the plasma in an inactive state. An injury or a trauma stimulates the platelets in the blood to release certain factors which activates the mechanism of coagulation. So coagulation is activated by breaking down of platelets. Certain factors released by the tissue at the site of injury also can initiate coagulation, that is thromboplasmogen, and the enzyme produced is called thromboplasmogenase. Calcium plays a very important role in clotting. What is the role of calcium? It is very important in conversion of prothrombin to thrombin. So thromboplasmogen produced by the injured tissue, thromboplasmogenase produced by the platelet. So they are all involved in this whole reaction. Any doubt? Anybody? Now we pass on to the second body fluid. I told you we are having two body fluids. First is the blood, second is the lymph. Remember, lymph is a fluid that is formed from blood and returning to the blood. Don't think that it is an entirely different thing. No, it is filtered from blood. Okay, and is returning to the blood. Now, let us see how the lymph is formed and how it is returned. Remember, blood is passing through the blood vessels that branches and become the smallest blood vessels called capillaries when they reach the tissues. So, on reaching the tissues, tissues are made up of cells. Okay. On reaching the tissues, the blood vessels are branching and forming very fine branches called capillaries. Capillaries are having only thin, flat, squamous epithelium. Their walls are lined by thin, flat cells called squamous epithelium. This epithelium is very thin. So this is the capillary. And blood is flowing under high pressure. Because of these two natures, what are the two natures for factors? One, blood vessel is very thin, lined only by squamous epithelium. Two, blood is under high pressure passing through that. So because of that, the liquid part of the plasma, okay, not all the plasma, liquid part of the plasma without some major proteins will be oozing out. It will be coming out. Plasma without large sized protein. Why large sized protein cannot come out? Because the gap between the cells are so small that large sized protein cannot come. But those proteins which are all dissolved, gases that are dissolved, substances that are all dissolved, they can pass through the gap between the epithelial cells. you are having small gap between the epithelial cells. So through that gap, under pressure, the liquid part of the plasma will be escaping from capillaries and they accumulate in the gap space in between cells. The space between the cells are called intercellular space. It is called intercellular space. So this fluid will be accumulating in the intercellular space, which is also called tissue space in brief. Okay. Now, some of the WBC can also escape. Why WBCs are coming? There's a reason for that. WBCs are having an ability to change their shape. So, they will produce small pseudopodia and then with the help of that pseudopodia, they will be changing their shape and thus escaping. So, by the special property of changing shape, some WBCs are capable of producing pseudopodia and squeeze in between the gap between the cells. That process is called diapedesis. It is not in your books. Diapedesis is the ability of amoeboid cells to squeeze in between, gap in between cells, capillary cells in the capillary wall. So such cells are also coming out, such WBCs are also coming out, such amoeboid cells are also coming out. And this fluid will be accumulating in the intercellular space. Thus, this fluid becomes an intermediary agent between blood and the cells. Whatever materials blood is bringing, like oxygen, nutrients, first will be entering into this intercellular tissue fluid. So, this fluid is called tissue fluid or intercellular fluid. And this intercellular or tissue fluid will be acting as an agent between blood and the cells. So, from this tissue gets oxygen, nutrients, etc. Tissues will be releasing carbon dioxide and other waste materials to this intercellular fluid. So, it is acting as a middleman or agent between blood and tissue. This fluid is called the lymph, tissue fluid or intercellular fluid. So, what is the composition of that one? It is correctly plasma minus proteins, without proteins. Okay, so plasma without proteins plus some WBCs, not all WBCs, some WBCs. That is the composition of this fluid. Plasma without proteins but with some WBCs is the correct composition of this fluid, which is the middleman, agent between blood and the tissue. Now you may have a doubt. As blood is passing through the capillaries, filtering is occurring and liquid part is gone. So volume of the blood is decreasing as time passes. But it is not occurring because the fluid is now returned back to the circulatory system. This intercellular or tissue fluid is collected by small capillaries. These capillaries are very, very small, which will be called lymph capillaries. These lymph capillaries join to form lymph vessels, larger structures called lymph vessels. These lymph vessels are just like blood vessels. Okay, they are carrying the lymph, taking away the, draining away the intercellular or tissue fluid. And as the lymph vessels are, see, these are all the capillaries. Large number of such capillaries are joining to form a lymph vessel. Lymph vessel will be having some swellings. This is the lymph vessel. They will be having some swellings called lymph glands or lymph nodes. These lymph glands or lymph nodes are located mainly in the neck, in the axillary region or in the groin region. Okay, we'll be studying about the lymph nodes or lymph glands in immunological system in next year topic. They are playing an important role in protecting our body from the attack of pathogens. They are immunologically very, very important. Why they are called glands? Because inside these lymph nodes, the T cells and B cells which are involved in immunity, we have studied. T cells and B cells, which are the lymphocytes, are all remaining, and they are all producing chemicals and different types of cells within the lymph nodes when they encounter with a pathogen. When they come in contact with a pathogen, when they come in contact with an antigen, a disease causing agent. So when T cells and B cells come in contact with the disease causing agent, the T cells and B cells will undergo multiplication, they will proliferate, that is the correct term. They will proliferate, multiply and produce large number of cells and chemicals. That is why they are called lymph glands. Okay, and they will be destroyed, the pathogens will be destroyed in the subsequent interaction. That is why lymph nodes or lymph glands are called filters of the blood, because they remove pathogens or disease causing agents which have happened to enter into the blood and intercellular fluid. When they are passing through these lymph nodes, they are all destroyed. Okay. Now, a large number of lymph vessels join to form still larger vessels called lymph ducts. You are having two large lymph ducts. What is that? One which is present on the left hand side, it is called thoracic duct. On the right hand side, you are having a thoracic duct. That's the most important, thoracic duct. This thoracic duct, which is on your left side, will be joining to the subclavian vein coming from the left hand. The vein coming from the left hand, it is called left subclavian vein. These are all, I told you, drained back into the blood. So, when the thoracic duct is opening to the left subclavian vein, the lymph is taken back, poured back into the blood. Similarly, blood from anterior part and the right side, they are all joining to form another large duct called right thoracic duct. Right thoracic duct. So we are having a, it is also called right lymphatic duct. It is also called right lymphatic duct. Okay. The right lymphatic duct is opening to the subclavian vein coming from the right hand. Okay. It is draining blood from the right hand. So blood again is receiving the lymph, and thus intercellular fluid, which is the tissue fluid, also called the lymph, is returning back to blood. This is the reason why there is no change in the total volume of blood in our body. It is filtered in tissues and returned into the veins continuously. This flow of the second body fluid, which is different from that of the blood but derived from blood. What is the main difference? This is having only plasma without proteins plus some WBCs only. That is the composition of the lymph. It is a colorless fluid, but it is having the same mineral constitution, mineral concentration that of plasma in blood. So both lymph and blood are having the same mineral distribution, so that there is no problem of water entering into the tissue or water going back to the blood. There is no such problem because the salt concentration is equal in both blood and the body fluid. Salt concentration, mineral constitution is same in blood and this tissue fluid. So there is no such problem of exosmosis or endosmosis. Clear? Now, what are the functions of this lymphatic system? Lymphatic system is having a number of important functions. Okay, the fluid present in the lymphatic system is called lymph also. Okay, lymph is a colorless fluid containing a specialized lymphocytes which are responsible for the immune response of the body, T lymphocytes and B lymphocytes, I told you. So it is having some WBCs. What is that WBC? It is the lymphocytes, T lymphocytes and B lymphocytes. This fluid is released out into the interstitial fluid or tissue fluid, has the same mineral distribution that in plasma. Okay. What are the functions? It is responsible for immunological responses. Lymph is a colorless fluid which is having an important role in immunological responses because it is having lymphocytes. It is having an important role. Okay, response for the immune responses. Okay. 2. It is an important carrier of nutrients, hormones, etc. I told you it is an intermediate agent. Okay. Exchange of nutrients, gases, etc. between blood and the cells always occur through the fluid. So, lymph is a carrier of nutrients, hormones, etc. 3. Our alimentary system, digestive system is digesting fat and converting into fatty acids and glycerol. These fatty acids are absorbed first into the lymph vessels present in villi of small intestine. Small intestine are having lacteals. Lacteals are lymph vessels absorbed through the lymph in the lacteals. Lacteals are structures which are present in our finger-like structures of the intestine called villi. We are having a large number of villi in our intestine, and into that villi, the lymph is forming a very important vessel called lacteal. That lacteal will be the first site to which the fat are absorbed. So it plays an important role in absorption and transportation of digested fat in the form of fatty acids. Okay, fats are absorbed through the lymph in the lacteals present in the intestinal villi. So with that, we finish the body fluids. Any doubt up to this level? Anybody? Anybody? Okay. Very good, Makale. Very good. What happened to many of you? I cannot see you, most of you. Only one, I think, was present. Is it because of a problem in the connection or any other problem? I cannot see most of you last week. Remember, body fluids are continuously being circulated in our body. There are two types of circulations, namely open and closed. Open type is considered as a very primitive feature, a very simple, which is seen in mollusca, arthropoda, etc. What is called an open circulatory system? Blood is flowing through all spaces inside the body and not through closed tubes called blood vessels. There are no blood vessels at all in the body. Blood vessels are completely absent. Blood is flowing through open spaces. So, wherever you are having cavity, even though it is a coelom, that coelom will be also filled with the body fluids, namely the blood. Such a type of circulatory system is called open type. Blood is flowing through open spaces and not through closed tubes called blood vessels. Now you understand what is a closed type. In closed type, you are having blood flowing through tubes. Only through the tubes they are all flowing. They are never flowing through open spaces. They are all flowing through tubes. These tubes are called blood vessels. So you are having blood vessels. It is considered to be more efficient, seen in annelids, vertebrates, etc. Why it is considered to be more efficient? Because as blood is flowing through small tubes, we can regulate the amount of blood given to each and every organ. If an organ is given large blood vessel, more blood will be going. If an organ is having only a small, narrow blood vessel, it will be receiving only small amount of blood. So, how much of blood is to be given to each and every organ? That is correctly regulated and controlled. And flow of blood is faster also in closed type of blood vascular system. That is why in all advanced organisms we are having a closed type of blood vascular system. All vertebrates are having a closed type of blood vascular system. Okay. So annelids and chordates have a closed circulatory system in which blood is pumped by a heart is always circulated through a closed network of blood vessels. It is considered to be more advantageous because flow of blood fluid can be more precisely regulated, controlled. Amount of blood circulated or given to an organ can be controlled effectively. Now, we are having different types of circulations. In fishes, a single circulation is seen. Vertebrates are all having a heart which is chambered, and the number of chambers in a fish will be only two. One, atrium. What is atrium? Atrium is receiving the blood, and there is one ventricle. So, in a fish, you are having only one atrium that is receiving blood from different parts of the body, which is pumping blood to the ventricle, and ventricle is pumping blood to the gills where oxygenation is taking place. Okay, after oxygenation in the gills, the purified blood is returned to the tissues. They are all taken to the tissues, to different organs. Okay, so you will be having pure oxygenated blood given to the tissues, and blood become deoxygenated because the blood is receiving carbon dioxide and other waste materials. Okay, now that deoxygenated blood is again returning back. So from tissues, the deoxygenated blood is again coming back. This is the situation in fishes. It is having only one atria and one ventricle, and blood is always flowing in one direction, only in one direction. That is from atria to ventricle, ventricle to the gills, gills to the tissues, tissues to heart, only one direction. Remember, only impure blood is always coming to the heart. Pure blood is directly going to the tissues, it is never coming to the heart. That is why Piscean heart of the fish is called venous heart. It is called venous heart. It is having single circulation. It is having single circulation. Whereas in amphibians and reptiles, the condition is different. In amphibia, in frog, we have studied in detail. Heart is three-chambered in nature. You are having a right atria, left atria, which are all opening to a single ventricle, heavy-walled ventricle. Blood from all parts of the body are collected by veins that are forming a sinus venosus, a common triangle-shaped structure called sinus venosus. Okay, that sinus venosus is the triangle structure located on the dorsal side of the heart, which is formed by the fusion of three important veins. Okay, and it opened by a single aperture to this is the sinus venosus, right atrium. Similarly, oxygenated blood, pure blood from lungs and skin are opening to the left atria, but both atria are opening to the ventricle. So pure and impure blood are mixed together. That's a problem. So pure and impure blood are poured into the ventricle. There is only one ventricle, so it becomes impure. Okay, mixed blood, and this mixed blood is taken to the lungs and skin for oxygenation through the truncus arteriosus. There is a blood vessel called truncus arteriosus arising from the ventral side. Okay. After oxygenation, the blood is returned. Okay, it is returned to the left atria through the pulmonary vein. Pulmonary vein. And again, blood is going to the different tissues and organs and returning through the three important veins. So here, there are two circulations, from heart to the lungs, back to heart, then from heart to tissues, organs, and again back. But blood that is running through these are mixed. So it is incomplete double circulation. Why you are having that term double circulation? Blood is taking two routes from heart to the lungs and back. So blood from heart to lungs and back. This is called pulmonary circulation. Pulmonary circulation is also called lesser circulation because it is only a short route. And the second circulation is from heart to organs, organ systems, and then back. It is called systemic circulation or greater circulation. But though there are two routes in which blood can flow, it is a mixed blood. That is why it is called incomplete. The two routes are not completely separated. Blood can flow in both directions in a mixed form. So it is called incomplete double circulation in amphibians. Similar condition is also there with minor difference in reptiles. What is the condition in reptiles? In reptiles, we are having incomplete double circulation, but in the heart there is a difference. What is the difference? You are having a pair of atria and a ventricle which is incompletely divided. Ventricle is incompletely divided. There are no sinus venosus, truncus arteriosus. Sinus venosus and truncus venosus are present only in frog. Here you are having veins bringing blood from different parts of the body. Okay, they are all opening to the right atria, and pure blood is taken from lungs to the left atria. Right and left atria are completely separated, but the pure blood and impure blood are mixed together in the ventricle. So again, it is a mixed blood that is circulated. Here also you are having circulation to the lungs and back, which is called pulmonary, and circulation from heart to organs, systems, and back. It is called greater circulation or systemic circulation. So here also in reptiles also you are having incomplete double circulation. Then you may say, in crocodiles you are having four-chambered heart. Only among reptiles, crocodiles are having completely divided, four-chambered. Still, in crocodile also, it is having incomplete double circulation because the blood vessels arising from the right side and left side are fusing together to form a dorsal aorta. So, there is always mixed blood that is circulated. Whereas, in warm-blooded animals like birds and mammals, the condition is entirely different. The ventricle is completely separated, so heart is fully separated to four-chambered. And so, there is no mixing of pure and impure blood. Okay, that is the situation in human beings. We are having complete double circulation, which we have to study in detail. Okay, now you understand. In birds and mammals, the ventricles pump it without any mixing up because the two ventricles are completely separated. The separated circulatory pathways are present in these organisms like birds and mammals. So, these animals are having double circulation. So, all vertebrates are not have the same type of circulation. Fishes, single circulation. Amphibians and reptiles, incomplete double circulation. Birds and mammals, complete double circulation. Crocodiles in there, definitely crocodiles are having a completely divided ventricle. It is the only reptile which is having completely divided ventricle. So within the heart actually there is no mixing, but blood is mixed because the blood vessels arising from these two are all joining. In reptiles that is the situation. Blood vessels which are all arising from these two blood vessels arising from the right and left ventricle are joining to form the main blood vessel called dorsal aorta. So impure blood, pure blood, they are all mixed here. Is that clear? There is one more reason. Can I tell it? It may be sometimes confusing to you. That is why I have not told you. At the point where these two blood vessels are crossing, there is a small opening called foramen of Panizza. Foramen means opening. Foramen of Panizza. So because of that, there is mixing of pure and impure blood. So always impure and pure blood are mixed and that mixed blood is flowing. Though there is no mixing of blood inside the heart in the crocodile. Crocodile is the only reptile which is having 4-chambered. Still in them also you are having incomplete double circulation. Any doubt up to this level? Okay. Now, we are all going to the study of human circulatory system. It is called blood vascular system because it is the body fluid, blood that is circulating through our tubes, vessels. So, we are having a closed type of blood vascular system consisting of a 4-chambered muscular heart. Heart is the pumping organ. We are having a large number of vessels that carry the blood. They are called blood vessels. Blood vessels are of 3 types. We will be discussing them. Arteries, vein, capillaries. So, arteries, veins and capillaries are the 3 types of blood vessels and the fluid which is circulated in them, that is the blood, which is the most important body fluid. So, blood, blood vessels and heart are the 3 main parts of human circulatory system. Okay. Remember, heart and circulatory system are derived from mesoderm. Remember, we are all triploblastic. We are having 3 germ layers, ectoderm, endoderm, mesoderm. And respiratory system is endodermal in origin. The circulatory system is mesodermal in origin. It is mesodermally derived organ situated in the thorax, in the gap between the 2 lungs. That gap is called mediastinum. In between the 2 lungs, there is a small gap called mediastinum. Please take it down. mediastinum. In respiration, I have told you these things. Again, I am repeating. Okay, slightly tilted to the left. It is having the size of a clenched fist. What is the size of your fist? Clenched fist. That will be the size of your heart. Okay. The rest we will discuss next week. Okay. So please read the book and study the class notes thoroughly. And if you are having any doubt, please clear it immediately. Okay. So before we start the session, you can ask me and get cleared all your doubts if you are having any. So it is time for me to wind up this session and say goodbye and good night.